the video materials. Definitely, maybe genetic information would be quite important too, but at least in that given example of epigenetics, the mother has a good behavior, then that traits somehow it's transferred to the offsprings. That was the epigenetic examples. Yeah, at least so far, the best known epigenetic modification is C methylation. That's the best known so far. Most of those C methylation happens in DNA. RNA is also modified by something, but it's at different positions with different modifications. So 99% of epigenetic markers in DNA is for the C methylation. Messenger RNA synthesis decreases. That's a so-called gene expression. Gene expression is decreased. And DNA, the replication we didn't mention, but that is also kind of suppressed. And euchromatin up the other component for matching to euchromatin is heterochromatin. Euchromatin is a more open form. Heterochromatin is a more condensed form. Open form is more the active for gene expression, and mRNA should be decreased. Methylation side is a back side rather than the Watson-Crick base pairing side. This is the front side matching to A. C, T, sorry, what did I say? It's A, okay. should be G. So C should match with the G to make a Watson-Crick the hydrogen bonding. And there's the front side. The methylation happens on back side. That's why even though methylation happens, DNA replication still works. Slow down, but still that is replicated. DNA double helix still can form, and still it can form the base pairing with the G. Okay? But the DNA replication can occur, reduced form, but still can occur. DNA methylation effect is quite clear. DNA methylation, gene expression down, okay? And histone methylation function sometimes up, sometimes down. Euchromatin increase means it's a gene expression up. mRNA increase up, mRNA decrease down, and histone methylation effect random. To some extent it's random. It's difficult to say in one word whether increase or not. Okay, this is gene expression of, right? Acetylation, histone, and DNA make a complex. DNA is negatively charged. Then histone would be negatively, positively charged. It should be positively charged. Then it neutralizes the charge of the DNA, right? So if histone is acetylated. Acetyl blocks the positive position, block the amine site. Then positive charge decreases. Then DNA histone binding increase or decrease? More positive, more negative, they bind strongly, right? Positive charge is decreased by adding acetyl group. Then interaction of histone to DNA decreased, becomes loose. It loose, then it forms more euchromatin. U means it's a real. It's a prokaryotic, eukaryotic. That U, karyo is a nucleus. Eukaryotic means real nucleus. Euchromatin means real chromatin, real DNA which does the gene expression. Okay, hetero means different. It's condensed and it looks different. So euchromatin increase. Acetylation make that binding loose. More open is RNA synthesis increase. Gene expression is up. Euchromatin is increased. Okay, so acetyl effect is clear in histone. Opposite to DNA methylation. Right? DNA is more methylated, down. Histone is more accelerated, it's up in terms of gene expression. Methylation pattern in DNA will accumulate all the time, but when we make a sperm or egg cells, still that remained in sperm or eggs. But when they fertilized, the majority of methylation is erased. More than 99%, almost erased, but some of them survive. And that methylation can be transferred to the springs. The majority of them is removed. The very small population of methylation pattern is survives and then transferred. Okay. That generates that epigenetic effect. Mainly transferred now, removed when still keep it, and that is removed during the development, and can be transferred a small amount, not a lot. Okay, this area, what do you call? This is coding area. Coding area has some exon introns in there. Intron is removed and exon is combined to make mRNA, right? So upper part here, for regulating the gene expression here, that part, what do you call? Promoter area. Okay. So there is an upstream, not downstream. 몇 시간 정도네? 1 minute. Okay, it's only CG sequence. So to make it easy to pronounce, we call postbit in between CPG. CG, one here, GC doesn't work, GC doesn't work, CC doesn't work. Only CG position is methylated, so only one position here. Okay, seems like you guys are studying quite well. And today we have 2 volunteers to present interesting work. So first one is Hyung-gu. Hyung-gu is going to tell us about a little more details and clear the information for how to select the hybridoma to make the monoclonal antibody. Move, please. Mic.
Okay. Hello, my name is Hyun-Woo, and this is a presentation about the hybrid-gamma selection in monoclonal antibody production. So before we get into the details, let's briefly recap how monoclonal antibody production is work. So first, the B cells or the spleen cells is isolated from the spleens and is fused with myeloma cells, which is a type of cancer cells. And the success rate of the fusion is quite low. So the selection procedure where we select hybridoma cells, the fused cell, from confused undesirable cells are needed. And only then we can proceed to select the optimal antibody that binds the target we want. And in this presentation, we are going to be focusing in hybridoma selection. So, as we all learned a couple of weeks ago, nucleic acid, among other things like proteins, lipids, or carbohydrates, is an essential molecule for life. So, nucleotides, which is a building block of nucleic acid, is critical for the cell's survival. Typically, human cells have two different pathways of synthesizing the nucleotides. One is the salvaging synthesis, and the other is the de novo synthesis. We won't be getting into the details of these two synthesis procedures, but we need to know about the two key enzymes that take parts in these pathways. One is the HGPRT, or hypoxanthine guanine phosphoribosyltransferase, which works a key role in salvage synthesis and use hypoxanthine as a substrate. And DHFR, or dihydrofolate reductase, works an essential role in de novo synthesis and use folate, formerly known as vitamin B9 or yoksan in Korean, as a substrate. In monoclonal antibody productions, myeloma cells that we use lacks the ability to produce HGPRT, meaning they cannot use the salvage synthesis to synthesize nucleotides and can only use the de novo synthesis. Now let's return to the original questions. How can we select only the fused cells? When the fusion is completed, there would be three types of cells remaining. First, the unfused B cells, unfused myeloma cells, and hybridomas, which is the cell we want. Unfused B cells will not be a problem since they have limited lifespan and will die anyway, which is the reason why we are fusing two cells in the first place. So that means the unfused myeloma cells would be the major issue. As I just mentioned before, they do not have the ability to produce the enzyme HGPRT, meaning they lack the gene. So they cannot use the salvage synthesis. Let's keep that in mind. And after the fusion is finished, we are going to culture these cells in an environment called the HAT medium. As the name suggests, HAT medium has three main components. First, hypoxanthine, which is a chemical that HGPRT used as substrate, and aminopterin, which is a structural analog to the folate and thymine. So aminopterin is a structural analog, so it acts as a competitive inhibitor of DHFR. This is how folate molecule looks like, and this is how aminopterin looks like. So as you can see, they are quite similar. The notable differences would be here and here. And thymine is just another material for salvage nucleotide synthesis. So as you can see, the HAT medium provides the materials for salvage nucleotide synthesis, as like hypoxanthine and thymine, but it inhibits the de novo synthesis of nucleotides by providing aminopterin as a competitive inhibitor. So, while unfused myeloma cells does not have the ability to use salvage synthesis, cannot survive, since de novo synthesis is inhibited. But hybridomas have the gene of the B cells, not the myeloma cells. So they have the ability to produce HGPRT, meaning they can utilize the salvage synthesis for their nucleotides, and thus can survive in the HAT medium. So a confused B cell just dies, and confused myeloma cells cannot survive due to the lack of salvage synthesis. So only the hybridomas survive and can simply survive. This was the procedure of selection in monoclonal antibody production. Any questions or discussion is welcome.
I have a question. Thank you for the presentation. Before the slide, you have the chemical structure of aminopterin and acromes. And first, immunosynthesis is working on the amine group. President pointed out amine group.
The reason why I said the difference is because they overall structure looks very similar. So this is actually the dihydrofolate, and DHFR converts folate to dihydrofolate into tetrahydrofolate. So this is the substrate, but this is not. Meaning it blocks the folate's way and does not actually take part in the reaction. Did that answer the question?
To make the DNA, to make the gene, you need either this one or this one as that. B cell has these enzymes. Myeloma does not have this one. Myeloma does not have this one. So to survive, you need to have either machine working, and here they have the inhibitor of this. This is the inhibitor for DHL. But even though B cell has this one, it cannot work. Inhibitor is active to everywhere. So you have a hybridoma, you have a myeloma, you have a B cells. So they should provide this machinery. B cell has both of them, but this enzyme cannot work because the inhibitor is there. So this pathway is blocked. Now only pathway you can use is this pathway. And then here they edit that substrate and put it on by the way, myeloma does not have this one. Only B cell has this one. So myeloma cannot survive. Only hybrid you can survive. B cell anyway you can live for long. So you will die not. So that's how you can select. This is exact the details of how hybridoma cell can be selected during those cultures. So in a sense, maybe this compound can be considered as antibiotic because it can block this enzyme, which is not necessary for human, but it's necessary for many bacteria. So combining of some of those genetic mutations to remove these and antibiotic, and that combination somehow make it easy to get small population of hybridoma out of the majority of the myeloma. So this is the details of the hybridoma forming and the hybridoma.
So first, I really liked your presentation because it was very easy to understand. And my question is that, according to your explanation, we use two types of synthesis, salvage and de novo. So, how would you know when the myeloma cells would have died? You just keep culturing them, and then after a certain time you just use them. Or, and another question is that, are there any adverse effects of using just one mechanism? So you're using, so normally you would use two synthesis methods, but you're using only one method for the hybridoma. So are there any adverse effects of that on the cell?
To answer the first question, you just wait for a certain amount of time. So, and that's just it. And the second question, no, this is, we are talking in a cell level, not in an individual level. So the only problem that might occur is the cell would not grow as much as we want. But I don't see any problem with that because as the name slightly suggests, the de novo synthesis is making an entirely new base, the nucleic bases from other materials, and salvaging synthesis is using the kind of decomposed parts of DNAs of other nucleic acid and making into the things that the cell can use. So most of the time, the salvage synthesis works predominantly than de novo synthesis anyways, so there would be little to no adverse effects. At least that's what I assume.
Okay, design-wise, this is just a design. So, like I said, this is a little more efficient because you can utilize some available intermediate, so like a chunky. So this one should be at the start from the very beginning, so it may take a little longer time and also more expensive. But if this is a live or die situation, anyway, the cell will take any of them to survive. So according to the design, it should work. But sometimes the cell finds some other way to save itself by making another mutation. Always there is a possibility that maybe the myeloma can survive. So if that happens, then you have a bunch of the cells you collected. You assume they are all hybridoma, so you can check whether they produce antibody or not. If they do not, then maybe some one possibility is maybe some myeloma they survive even though they are not fused with the B cells. That's always possible stuff. That's kind of mechanism of antibiotic resistance or resistance of anti-cancer drugs. Lipoforms, this is just we utilize the information we know and we design, but sometimes they can escape. That's always possible. So that's why you need to check the quality of the hybridoma collected, whether it's pure or whether it's efficient or not. Second one, side effect. As you mentioned, this is a cell is extracted from the body. So this is the test tube. If the experiment is happening inside the body, we call that experiment as in vivo. Vivo means it's the liposomes. In vivo cases, adding this kind of inhibitor and extra nutrients can be potentially dangerous. But once it's taken out, then fully in test tube, we call it in vitro. But this is kind of in the middle. The cells are still alive, and then we took out from the body and then do the experiment outside of the body. That's what we call in between, in vivo, in vitro. So this experiment we call ex vivo. We took out some component from the vivo, but we do the experiment outside of the body. So even though something happens to the cell, we are just using these cells as a tools, production factory. So if we can make antibody, that's fine. We can extract the antibody and use it. We discard those leftover cells. So that's how we control. Okay, thank you. Okay, we have another presentation today. Very volunteered to give interesting examples for epigenetics. Please. Mike.
Hello, I'm Yeren Lee, and I'm going to briefly talk about epigenetics.
Slow.
And sorry, but I'm a fast speaker. I'll try to speak as slowly as possible.
Let's set up we're on the speed as a 0.7.
Okay. So, in 2003, the Human Genome Project was completed. And starting this project, scientists thought that if we know the whole sequence of genes and their locations, human beings could overcome several diseases like cancer. However, today in 2023, we still suffer from diseases, and there wasn't any dramatic changes. So why can't we solve these problems? It's because the sequence of our genes is important, but the expression of these genes are more directly connected to diseases. So, epigenetics is the study of changes in gene expression. These changes we talk in epigenetics are reversible and does not change the DNA sequence. And they are affected by our behavior and the environment. Let's look at a simple example. We studied about honeybees and their sex decision last time. And we studied that the queen bees and female worker bees are sisters. However, queen bees are bigger and they have a longer lifespan, and they have different roles in bee society. So here's the question. How is the queen bee decided? Queen bees doesn't have special DNAs, and surprisingly, the difference in the food they eat when they are in larva stage makes the difference. All the honeybees are first fed with royal jelly, but only the future queen bees continuously eat royal jelly and others don't. So we can say the food, the environment the bees are laid in makes the difference of their shape, size, and their roles. It is known that there are three typical phenomena in epigenetics: DNA methylation, histone modification, and RNA interference by RNAs like microRNA or siRNA. But since we saw this in the video, I'm going to just skip it, and here's the summary. So when DNA is methylated, gene expression is suppressed and it is catalyzed by the enzyme DNMT. And when DNA is demethylated, gene expression is induced. When histone proteins are acetylated by histone acetyltransferase HAT, gene expression is induced. And when it is deacetylated by histone deacetylase, gene expression is suppressed. Now let's look at the honeybees. As I said before, the difference in diet makes the change between queen bees and worker bees. And scientists say that honeybees are one of the most representative examples of environmentally driven phenotypic plasticity. There's an interesting experiment with this. Well, scientists first fed the larvae with worker bee diet, and also they knocked down the DNMT genes. DNMT is the enzyme that methylates DNA, so when DNMT gene is knocked down, DNMT can't do its job. Surprisingly, even though they ate the worker bee diet, 2/3 of the larvae grew up to become queen bees. By knockdown of DNMT gene, some DNAs couldn't be methylated, so that genes were expressed. So we can predict that these specially expressed genes induce the larvae to become queen bees. And we can also predict that in royal jelly, there is something that works similar with a knockdown of DNMT, something that inhibits DNMT and inducing expression of special genes. Actually, it is also known that in royal jelly can activate histone deacetylase inhibitor. So when a larva eats royal jelly, acetylated histones are not that much deacetylated, inducing special genes to be activated. And there are more examples. As you can see, there are two shapes of water flea, 물벼룩, and one has something on their head and one doesn't. The thing on their head is for their protection from their predators. Surprisingly, for water fleas that lives in a place where they can sense molecules that their predators have secreted, they form as the upper form. But the water fleas that lives in a safe place where there is no marks of their predators, their appearance is like the lower form. And there is an interesting experiment with water fleas too. So everyone would definitely expect that their offsprings will have the same shape as their parents. However, when we change their habitats, the initial generations kept their ancestors' shape, but as time passed, their shape adjusted to the environment. It's very similar to the example that we saw in the low liquor mouse and high liquor mouse in the video. Actually, there are numerous examples in the animal kingdom. Many insect species initially change their temperature to control the development of their offsprings, and in reptiles, 파충류, the temperature is very important for their sex determination. And desert locusts, 사막 메뚜기, can change their shape depending on their density of their population. The change of larvae into butterflies and moths is also dramatic changes from numerous change of DNA and histone modification and manufacturers of microRNAs. Now you can wonder, what about in humans? Are epigenetics that important? Well, epigenetical processes are very important to our lives, and it has happened even when we weren't even born. For example, X chromosome inactivation is a very important epigenetical phenomenon. But since Professor explained about this, and so I'm gonna kind of skip it and tell you about other examples that you might think that it's more related to our everyday life. Well, we all know that smoking is bad. It causes cancer. However, in detail, there's also differences in epigenetical pattern between smokers and non-smokers. So, when nicotine first comes into our body, it binds to nicotinic acetylcholine receptors, and it leads to increase in calcium levels. This results in downregulation of DNMT1 gene and upregulation of DNMT3B gene. So I didn't mention this before, but there are several types of DNMT, and DNMT1 is the DNA methyltransferase that attach the newly made strand during DNA replication. So DNMT1 is an important gene on maintaining the original DNA methylation pattern. So, and DNMT3B is the DNMT that makes the brand new methylation patterns. So, when downregulation of DNMT1 happens, the genes that were usually not expressed could be expressed and activated. And when upregulation of DNMT3B happens, the genes that were naturally not methylated could become methylated. So by smoking, some genes can be overexpressed and some could be hypermethylated and not expressed, eventually leading to cancer. There are many ongoing epigenetic experiments with identical twins. Since their genome sequence is 100% same, it's relatively easy to look at epigenetical changes. And it is known that 70% of epigenetic patterns are determined right before and right after birth. And it means that the left 30% changes over time. When identical twins are first born, their epigenetical patterns are almost the same. However, as older they become, epigenetic patterns differ. It means that humans interact with the environment they are laid on and changes the gene expression. The things we eat, frequency of exercising, how much we smoke, how much we drink, it is directly connected to our health and epigenetic changes. Your inborn genome is important, but it's not all. Thank you for listening. Are there any questions?
Any question?
I was wondering about the different kinds of DNMT. Like is there some use for DNMT3B, or is it like just a bad thing?
I think it will be good because we have to adapt to the environment. So to adapt to it, sometimes we have to regulate some genes, and I think that's the use of DNMT3B.
So about the multiple types of DNMT, the experiments about the 3B and knocked out the DNMT, the researchers knocked out every types of DNMT, or did they choose a specific subtypes of DNMT?
As long as I understand it in this paper, I think it was DNMT1. So when the cells replicate, the DNMT1 roles for methylating the original patterns. So I think it was DNMT1. They knocked out DNMT1 enzyme gene.
Let's just think about the meaning of maintenance. Let's say here is the gene, and here is the methylation here, and this is the 5 prime, and this is 3 prime. We can make a copy of the DNA. And DNA synthesis should be out of this way. So this is a template. Synthesis should happen from 5 prime, then from here, CTTTP, and CG. So this is a new strand synthesized. Then this does not bother, this methylation does not bother for arsenic group is pairing. So G can come here. And this G would C here. Now DNMT1 may come here, recognize this one. Oh, methyl group here, and I want to methyl group here too. Then it has the same pattern of methylation. Once we divide it, make one copy, same methylation pattern will remain. So this is the maintenance. Now, new methylation pattern is maybe more complicated, and there is some form of DNA conformation. It's a complex together with the proteins. And depending on environmental changes, and then recognize some of these three-dimensional changes, and then methylate the new set. That's something we can do. So this general methylation pattern, statistically where and how much may not be that clear. So how actively, how selectively, how fast, how slow may depending their roles are a bit different. That's why this observation is in general, PMNT was decreased, then, and the result is what? Gene regulation up or down. DNMT is decreased and gene expression is up or down. It's up, more open form. Then we have several things. DNMT, DNA methyltransferase, and we heard HAT. This is the histone side, right? Histone deacetylase. So these three enzymes has roles. If this goes up, gene expression goes down. If this goes up, gene expression goes up. If this goes up, gene expression goes down. General trend, DNA case methylation, histone case methylation is kind of clearly defined this way. If we have DNA, if we remove this part, the whole DNA part, then we call this not out. Take this out, so remove the gene, then definitely gene expression will not happen. If you insert new gene here, then starting from here, we call not in. Okay? This is a genetic change. Genetics is changing, DNA sequence is changing. Insert in or take it out. Now we are talking about not touching the sequence itself. Expression is up or down. So that's why utilizing this, still we want to say something. If this goes out, then gene expression down. So this one is what we call it. Now, down. There is no official term for this. But in comparison with this, maybe meaning-wise, this can be called as a knock up. This is the gene expression down. This is the gene expression increase. Maybe we can call knock up. Those are different from this, right? This one is a genetic change. Sequence is inserted or removed. This one sequence is still there, same sequence, just regulation into change. That is the key part of epigenetics. Okay, thank you. Okay. Today, since we had and I asked you some interesting examples of epigenetic examples, there's a lot of good answers here. So Jagan mentioned about this induced pluripotent stem cell, blah blah blah. This is a topic of next week. Okay, we skip. And here, CPG. Okay, this is about the aging clock. If you read the DNA methylation pattern, we can estimate the aged status of the cells of the individual. So this topic will be handled in aging the part after the midterm exam. Okay, another interesting example here. So this mouse had the agouti gene. Then depending on what kind of the nutrient they get during their development, and eventually they change the fur color. And that pattern is regulated by DNA methylation cofactors, like a vitamin B6 or vitamin B12. The folic acid is also one of them. And depending on those DNA methylation pattern, more DNA methylation happens, then they becomes more black color, more healthier. More poor nutrient, they becomes more yellowish color, and then they have more chance to get the disease. Kind of observation is also found. In comparison, in cat, there's another, the color changing, the pattern was discovered. So Yerim briefly showed those X chromosome. One was with the red color, one was small dot there. So female has 2 copies of X chromosome. Male has only 1 copy of X chromosome and very small Y chromosome, right? So that's not a fair comparison, fair competition. Okay, female has a double amount of a gene. Male has only 1 gene, and that small part is mainly regulating those matching side of X chromosomes, the function here. So if you have 2 fully expressed and only 1 X expressed, male cannot compete with the female. That's not fair game, right? That's why in female case, out of these 2 X chromosomes, 1 is deactivated. And that deactivation seems like deactivating pathway is like by methylating those one of those X chromosome, and then it's methylated and deacetylated. Those 2 things happen together, and they are condensed further and further, make very tight the X chromosome. That was the picture what Yerim showed. So one was the A is active one has dispersed form. The other one, inactive version, is a condensed form like this. And that happens, that one of those, the gene of the fur color is on X chromosome. So in cat, this color changes. So this side is activated one, this is inactivated one. This kind of the cat color difference, most likely you may see only in females, not males. And this looks like exactly half doesn't happen. It's almost random. There's X inactivation in all of the body. It's not only one side. So looks like it's random. So which X chromosome will be deactivated? It looks like random. That's why we see this kind of random patterns. And if it has the same color, most likely this is male, this is maybe female. Okay, so that's the one example of the epigenetic modification happening in 2X situation. And there's a little more questions. Maybe later.
Professor?
Yeah.
Jenny has a question.
About the cat, so is the colors like random on the cat because the X inactivation is different in different cells?
Right.
Right. Right. So it's like cell by cell, basically.
Right.
Is it similar in humans?
Yeah.
So is there some kind of similar effects?
Not apparently showing off, but seems like it's not really cell by cell, it's kind of chunk by chunk. So maybe one cell is decided to deactivate X1, then X2 expressed and they proliferate and make some small colony here, and X1 colony here. It's kind of a mosaic structures or chimeric structures. Okay, methylation in histones, yes, it's already explained. In DNA case, methylation effect is a knockdown effect. Looks like I just referred. The histone case, there are some more modification. Histone is a protein. Protein has 20 different amino acids. DNA has only 4 bases, right? ATCG only 4, so it's simple to choose one of them. Protein has 20 different sites. There can be acetylation, methylation, phosphorylation, ubiquitination, and carbohydrate addition, and many modifications can happen. That's what we call post-translational modification. After protein is synthesized, the different position is modified with the different groups. And their effect is difficult to judge all as a one trend. Sometimes upregulate, sometimes downregulate. It's kind of a mixed case by case. Okay, this is the question. So X chromosome is like this, and then now that means only one X chromosome survive. Then if that gene happens in other area, then what's going on? Is that only dominant versus the dominant gene versus the overpowers of a recessive one? This happens by only one gene is expressed and the other one is not? Maybe not. So in X cases, X1 and X2, only one of them survived. So depending on those genes, the only one gene dominant. But in other cases, that's too abrupt and to make that kind of decision. So in this case, let's say activated one, the dominant one and recessive one, it may be more balanced. Capital, 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 small, 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 maybe 3 levels. So it's a more active one and medium one, and then at the very recessive one. That's more common cases. So in that case, maybe red color, white color, maybe pink color in between kind of a situation. Sometimes it's very dominant of the dominant phenotype, but in many cases it's mixed up in between. So that's kind of the observation we have. Now, even one phenotype, time to time, it's difficult to say which one is the more dominant than the other. Now, the question is, melanin in human, it has several alleles. How does it work? That's just a more complicated problem. More factors than maybe whether they are working together or controlling to each other. That's maybe just make the situation a little more complicated. DNA cytosine methylation is like RNA uracil methylation to make a thymine. Then why this happens? Why this one is chosen that way? Well, that's a good point. So here's the DNA. And this is, it has a U. But if you see those methylation site of the C is here. This is what's encrypted base pairing side. And then this is the back side. This methylation site is almost the same over this T position. Right? And DNA chose this T, and RNA chose this U. Then why this one is not participating in the regulation? Only C participating in the regulation. Well, I would say that may be almost randomly chosen during the evolution. There is no special reason why this should be T and why this should be U. Even if you change this, still that works. Still the replication still happens. Similar to the methylated cytosine, even though you change it with this one, still DNA replication happens in in vitro system. So that means if you think about this is ATP, GTP, this is CTP, TTP. So if you look at these cases, usually we hear biological battery, ATP. But if you look at those chemical storage, energy storage part, this is the energy storage part, right? Then why only ATP? The others are just regulating all the other roles. Maybe no specific reasons. Maybe ATP was chosen in the beginning, and ATP was chosen, and it just dominated the enzyme of the functions. So it's almost like a random choices here. Similar things happened, we can assume. We don't know exactly. So this one has been chosen, both sides is the same, and methylation they chose, nature chose as regulating part. That means maybe binding site, the binding event of an enzyme to this site is somehow different from the open site. And then that makes those situations gene knockdown rather than these cases. Okay, another question. Okay, zygote forms and the blastocyst, DNA methylation happens to almost all genes. And this one. So when the sperm was made, so this is methylation percent. Methylation percent average is around here. And sperm cells still have good amount of methylation. Egg cell is a little less, but also they have a methylation patterns. So that methylation pattern is derived from the original father and mother. And when they meet, and then the sperm, very quickly, the DNA methylation is erased. Almost the bottom to go to the bottom. mother side egg cell is doing a little trick, and it keeps some methylation pattern to some extent, even up to the blastocyst side. That means methylation pattern of egg may have a more role to control those embryo growth. Right? So that means the genetic information of father and mother may not be equally contribute. At least the very early stage of this, the embryo, the development, their involvement may be quite different. So it reaches almost the bottom here, but that's not fully 0%. There is some genes that survive here. That's why some of those DNA methylation pattern still survive and transferred to the offsprings. And one of the questions, how this happens, avoid from being, well, that's a more technical part. And we can assume, we can imagine what's going on. Maybe some of those, the area may be more protected, more folded, and then does not allow to access the demethylation enzymes, possibly. Okay, another question? Okay, if we need like a protein legs and then we need to make more protein, and then how does the HAT can be turned on, distinguish the genes and which one goes up? Well, if you think about those, the DNA, the gene expression, maybe this method, using HAT or HDAC or DNA methylation, this is a very broad scale. Once it is activated, it can methylate this much and deacetylate this much. It may be very difficult to control one by one, those. So, usual cases, this kind of the big landscape, rather than this approach, they may choose transcription factors and maybe other proteins and recognize those as specific parts. That's kind of small changes all the time they can do. That's normal situations. Epigenetic modification will happen very big scale. Okay, based on this, so one example here. I don't know whether, do you know this person? At least do you know that the name and this person's, I don't know whether you have seen her movie, but she was very famous the actress and very beautiful, and until quite the aged the stage, she kept almost the same slim the body line kind of things. Many women liked that kind of a phenotype, but actually that was derived from this very difficult timing just before the Second World War is ended. So in Netherlands, there was a severe famine situations, short of the food. And all the populations, almost for 2 years, they were highly restricted from the food. And it seems like that effect generates significant impact to the embryos. And this is systematic, not designed, but this is recorded and then could be followed. And then they found that some area here, very early stage, up to 3 months, if the embryo were not properly provided with the nutrients, then that impact goes almost the whole lifetime. She was one of those victims. And that kind of the thin the body and also less developed the status, even that could be transferred to the next generation too. Another interesting point is, if the shortage of food, that has very strong impact only in this period. If that happens in the later stage here, that doesn't affect much. That means maybe during this period, those epigenetic markers like a methylation or demethylation pattern, it can affect much stronger here, and the others are almost like this is just moving forward for just a maturing stage. So that's why we hear many times that very early stage of the pregnancy, you should be very careful not to expose them to the harmful materials like smoking, alcohol, and take the folic acid. Folic acid, 엽산, folic acid is the strong methylation cofactors. Do not allow too open of the gene expressions. So more regulation is safer for this period. So this is kind of a historic observation of epigenetic impact. And we have seen this. So genetics versus epigenetics, this is sequence changes. This one is without the sequence change, still we see that impact. And that's why this is, the epi means is above. Genetic sequence is the same, but there's another regulations. Let's just say this is genetic, the sequence, then epigenetic is more like how to operate this, which part we emphasize further than the other. So one part we have already learned, the DNA methylation and the histone modification. When I was a student, so this person, Darwin, and this is Lamarck, and we learned with this kind of examples, which one is the correct one, whether this getting longer of the neck is by habit and the transfer of those habits to the next spring, the offsprings or not. We learned that Darwin is correct. More diversity generation and selection is the explanation of a genetic impact. But now with epigenetics, maybe he was not fully wrong. So his impact may have some impact because these habits, it can be transferred to the next generation too. So sometimes we believe that one is the fully correct, but in many cases, these changes and mutations, sometimes it doesn't match to each other. So you have seen these examples. Even though genetic background is different, whether you have good mother or not, that makes the character of offsprings even to the next generations. So this one we practiced already. This is the promoter area. This is a gene coding area. This is the 5', this is 3'. Okay, so if that maturation happens, so this is open form, open structure, and this is a more condensed form. So this is euchromatin area. This is actively ongoing gene expression site. And this one is kind of a blocked, chunk by chunk. If you use this HAT, HDAC, DNMT, then you do not just block one gene, you block one chunk each. Real image is like this. If you look at the cell, this is the heterochromatin area, block ones. This is the open site. So this is a relatively very actively expressing site, expressing the gene expressing cells. If it goes more suppressed site, then you will see this dark area is bigger and bigger here and there. Okay? Okay, any question for the epigenetics? I have several more slides, but we can handle this later. Genomic imprinting, we took a little example in terms of the inactivation. Okay, if you have a chance, then I would recommend again this book. It's quite a good book to get more examples and interesting stories. Okay, so next week we are going to study using this concept into stem cell and artificial stem cell. Okay, any questions for epigenetics?